Welcome to the first chapter and first lecture of Biology 100. In this chapter, we will cover scientific thinking. First and foremost, you are a scientist. Whether you thought that before this class or not, you have been using scientific reasoning to make decisions as well as to figure things out in your life, like maybe why did you not get enough sleep last night? Why are you tired this morning? Why does your stomach hurt? Why are you feeling sick? Things like that. Because when it comes to science, honestly, all we're doing is trying to find answers to questions about phenomena that we experience. So scientific thinking and biological literacy, which we will give you a definition here in a bit, are essential in the modern world. So again, we are going to answer a few questions here of what is science, what is biology, and essentially just start asking questions about the world around us. And these questions are going to help us answer that. First and foremost, science is not just a body of knowledge. It's not just a list of facts to memorize. It's going to be a process, specifically an intellectual process that's going to encompass observation, description, experimentation, and lead to an explanation of the natural phenomena that is happening around us in the world. In other words, science is a pathway by which we can come to discover and better understand our world, and it's not just our world anymore, actually. We are able to look at moons of other planets as well as other planets and other solar systems. So we are using science to figure out the phenomena not just happening on our planet, but also in our solar system, our universe. On a smaller scale, when we think of science, it is a way to put products to the test in some ways, to test a claim of either a product, maybe a politician, maybe a celebrity or an influencer, or maybe just your great Aunt Betty. It is important to question these claims, otherwise you might believe something that isn't true. In this case, I have two products for you. We have Cocoa Krispies and a product called Airborne, which is an immune support supplement. Now, both of these products are making the same claim that they help support the immune system. Your immune system is the part of your body that is going to help defend against pathogens, whether it's bacteria or viruses, things like that. Now, if we use scientific reasoning, you probably immediately looked at the Cocoa Krispies and kind of squinted your eyes and said, I don't think that one's telling me the truth. However, the other product says it has vitamin C, there's an orange on it, and you probably look at the packaging and see that it looks kind of adjacent to medicine. Now, congratulations if you figured out that Cocoa Krispies was probably not telling us the truth, but neither is the Airborne product. Now, I'm not saying that there are some good things potentially with these products. However, there is not enough data that supports either product in their claim that they help the immune system. So this is how scientific thinking and questioning whether or not a product or a person is telling us the truth, and we can put these claims to the test. Through the semester, I'm going to try to instill this ability to think scientifically into you so that you have this for the rest of your life. However, the purpose of the class is actually to talk about biology, and if we look at this word, bio and ology, we can break it down into its definition. Bio means living. Ology means the study of. So when we smush it together, we're just talking about the study of living things. So thinking scientifically can fall under this category of scientific literacy. This is a general evidence-based understanding of the basics of science. So in general, as I said before, it's just a way to think scientifically, using knowledge that you gain to make wise decisions, both for yourself and also to benefit society. This is very important, especially nowadays when so much social media is coming across your screen and trying to influence you to I don't know, buy a product that's supposed to be healthy for you, a better way to lose weight, maybe about something bigger like climate change or whether or not to buy a product that tells you your ancestry. Either way, this is incredibly important and will continue to be so. And what I want you to start doing is having a healthy dose of skepticism about pretty much anything that you hear. Now, that is to say, you should question claims. You should question those big, too good to be true products or hacks, as some influencers might call them. Skepticism is really important. That just means that you are questioning the things that you're hearing. I like to say that when it comes to science, it is not something I believe in, it's something I know. And that is because science is empirical. There is a physical basis to everything that is scientific. So, scientific thinking, in and of itself, that ability to think scientifically, is based on physical experiences, physical observations, both of which are going to be rational, testable, and repeatable. Otherwise, it's potentially up to chance that the phenomena you observed occurred. We'll talk about that more a little later. Scientific thinking, equally as important as being based on experience and observations, is also self-correcting. There's not a single one of you listening right now that you have not been wrong at some point in your life. I do hope that at some point when you were wrong, that you changed whatever it was you were doing that was incorrect, or you grew from it. That is something that is integral, very important when it comes to scientific thinking. It is self-correcting. There is an advantage to be able to change your mind if it turns out you are wrong. Now, the next term, kind of going in tandem with scientific literacy and scientific thinking, is biological literacy. This is going to be a process where you can use scientific inquiry, so questioning, to think creatively about real world issues or maybe products or whatever it is in front of you, and be able to communicate those thoughts to others. And once you find out more information about what you are questioning, you are going to integrate those thoughts into your decision making, which is honestly the hardest part. I like to tell you guys about palm oil at this point in time because we will talk about it again near the end of the semester. The harvesting of palm oil in the rainforest is one of the biggest reasons that the rainforests are being destroyed. In the last 25 years, more than half of the rainforest has been destroyed, and unfortunately, guys, that means it's not going to come back. Now, if you would like to put that decision making where I hope your heart should be, that we don't want to destroy the rainforest and that biodiversity that is very important to the planet. We'll talk a little bit more about that in the next couple chapters. Not buying products that contain palm oil is the best way to do that. By putting your money where your heart is, that is the best way to ensure that those products potentially change and stop using palm oil, and speaking out about it, as well as communicating to others what is in these products and what it's doing to the planet. Again, that decision making is the hardest part because, let's be honest, a lot of the products that have palm oil are delicious. We like to eat them. They're usually snacks that are very integral to us. Maybe whether it's, for lunch or in a vending machine, or you're just trying to grab a quick bite, whatever. Palm oil is in so many products, and it makes it difficult to make that decision, even though you know it's not good for the planet. If you were present for the first lab, the information in the next couple slides should be very familiar to you. It is the scientific process or scientific method. Now, the steps might be in a slightly different order than what we talked about in lab, however, it's pretty much the same information. For step one, we are making observations. So this is something you're probably going to do with your peepers. You're going to look at something, observe something, notice something about it, and start asking questions about the phenomena that you are observing. For example, maybe we're looking at a plant and we notice that, gosh golly, I've already noticed this forever, but the roots grow down, but I don't know why. How does a plant know, even though it doesn't have a brain, which way is up or which way is down? How does it know to grow the roots into the soil and down, and the stem and the leaves up above the surface of the soil? This is going to lead to a handful of questions. Probably the most important questions you can ask are going to be causal. Make sure you know that that word is causal, not casual. Causal questions will lead to a hypothesis that can be tested, as well as predictions can be made from this hypothesis that is stemming from these causal questions. Now keep in mind that this term causal, we're meaning cause and effect. So for example, after our observation of our plant growing into the ground, we ask the question, does gravity play a role in a seed cell knowing which way is up? You have other questions you can ask. These can lead to other interesting discoveries, but they're not answerable by a hypothesis. So these are described as descriptive questions. So from our observations, we are able to form a hypothesis. In this case, the example of the hypothesis we're going to write is seeds know which way is down due to gravity. A hypothesis is going to clearly establish alternative explanations for the phenomena you observed, as well as generate testable predictions. Something I want to get across is a hypothesis is explaining that there is a relationship between this thing and this thing, or there is not a relationship between this thing and this thing. Either way, we are describing something affecting or not affecting another thing. In this case, we are saying seed growth is affected due to gravity. There are two types of hypotheses. You have an alternative hypothesis and a null hypothesis. The alternative, make sure you know this, is the positive statement of the hypothesis itself. Gravity does affect direction of root growth. That is a positive relationship. One thing is affecting the other thing. The null hypothesis is going to be the negation of the positive. It is going to say something like, gravity does not affect direction of root growth. There is no relationship between this thing and this thing. Remember, a hypothesis is describing a relationship or the lack of a relationship between two things. You are going to typically use the word affect or not affect. Once you have your hypothesis, you are able to then make a prediction. The most important thing here is that a prediction is an if-then statement. This means that we are making an estimated guess over the specific outcome of our hypothesis. If gravity affects the direction of root growth, so that is a restating of the hypothesis, then we should see atypical root growth in microgravity. All we're saying with our example here is that if we put plants into anything other than what is normal gravity on Earth, we should see the roots do something weird. They should grow in a weird pattern. After you have made your prediction, we're going to conduct a critical experiment to test the hypothesis itself. For the example that we've been talking about, what we would do is potentially send some seedlings up into the space station and test the hypothesis, see how our plants grow in little to no gravity. For the final step, we draw conclusions and potentially need to make revisions. The biggest thing we're looking for is patterns and relationships that are going to help support the evidence for our hypothesis. That is the end-all be-all goal. Now, half the time, probably more than half, you're going to find that the patterns and relationships that you find might not support your hypothesis. That doesn't inherently mean that you're wrong, it just means that that's not the right answer. And it's okay to take the information from that and go a different direction. Maybe you just rework into a different hypothesis, and you just keep going from there. It's not that you're wrong. It's that that answer was not the most correct answer. And for the record, I know I don't have a picture or anything showing it, but if we did grow seedlings in space, like the space shuttle or space station, then what we would see is weird root growth on those plants. And that does support that hypothesis that gravity is affecting whether or not a plant knows which way is up or down. There are two terms that are usually used interchangeably, which hopefully after you learn this, you'll make a decision not to use them interchangeably. A hypothesis is different than a theory. Hypotheses are, a proposed explanation for a phenomenon before you do any testing. A theory is not a speculation or guess, it's a broad hypothesis that is supported by many years of data and research. It's repeatedly tested, and it's also not something that you guess before you start collecting the data. It's usually widely accepted as well. There are three theories that I would like for you to write down as an example. The first is endosymbiosis. We'll be talking about that in chapter 4, as well as the theory or cell theory. That's that third option there. That is also in chapter 4, and we will eventually talk about evolution. Many of the theories I have written up here, however, are based in physics. And that is not necessarily my area of expertise in any capacity, but if you do have questions, please let me know. When we conduct an experiment, there are a few parts of the experiment that we want to make sure that we understand. The first part is the treatment. This is going to be a condition that is applied to the experimental group. The experimental group is going to be the group in which that is given the treatment. There is a third very important component when we talk about experimental groups and the treatment, and that is the control group. I'll give you an example here in a little bit that explains why control groups are so important. But it is a group of individuals who are treated normally and identically to the experimental group minus the treatment itself. It is and I would write this in here, baseline. It is a baseline so that we know if there's any difference between the control group and the experimental group, that the treatment is doing something, that it is causing some type of change, whether that means it's working or not working. It just depends on what the treatment itself is. You have to have a control group. Variables within our experiment, and probably terms that you've heard before, especially if you were in lab earlier, is independent and dependent. Independent variables are the variable that is controlled in the experiment. In our plant experiment, it was gravity. In the dependent variable, that is the variable that responds to the experiment and is measured. In our experiment from before, that would be root growth. So the independent variable will be independent of the dependent variable, whereas the dependent variable depends on that measurement that is based on that independent variable. Another very important feature of a good experiment are the controlling variables. This is a way to minimize differences between the control group and the experimental group other than the treatment itself. This is going to help us have confidence in our results, especially if there's a difference between the two groups, meaning that the treatment does have some type of effect on our subjects. This is very important, potentially leads to a less bias in the data, and again, more confidence that we can support our hypothesis or not. Now, this example I have for you is a flawed experimental design with no controls. The first time that this was studied was in the late 1950s. A doctor was studying how to treat gastric ulcers, where gastric ulcers are a type of wound that opens up in the lining of the stomach. And due to the exposure of stomach acid, this causes a lot of discomfort. It can lead to a lot of nausea, potentially vomiting, maybe blood in the vomit or stool. Either way, it's not a fun time. So he took 24 individuals and he put a balloon in their stomachs, and the balloon contained a refrigerated fluid, and funny enough, 100% of the patients experience relief from that, the cold balloon. However, there is an issue with this design. There was no baseline. You need a baseline in order to determine if the treatment itself actually has an effect on the condition. So, a few years later, there was another researcher who looked at this research and said, I think this might not actually work. So instead, he took 160 individuals, which is a substantially higher number than 24, and he divided between the two groups an experimental group and a control group. The experimental group, composed of about 82 people, received the cold balloon. The other 78 received just a balloon that had room temperature fluid. Now, what we find is that there's little to no difference between the individuals that received the treatment, the cold balloon, versus the individuals who just got a balloon that was room temperature in their stomach. That tells us the treatment doesn't work, that there is no difference between the individuals of either group. That means the treatment to treat a gastric ulcer with cold temperatures does not actually help an ulcer. It does not relieve that discomfort. In fact, the leading hypothesis was that consuming spicy food is what leads to gastric ulcers. However, we know that's not true now, but there was a lot of good thought behind applying cold temperatures to an area that feels like it's burning. However, it just does not work. When it comes to some treatments, we have to look at medical studies. And unfortunately, we see this condition called the placebo effect. The placebo effect is a phenomenon where pretty much anybody responds favorably to any treatment, whether it's the experimental treatment or whether or not they're in the control group. However, this can be quite unfortunate when it comes to medical studies, maybe for cancer treatments, where the subjects do not know if they're receiving one treatment or they're in the control group. Now, because they think they're potentially receiving this medicine, we do start to see the individuals become healthier, maybe more active, start feeling better for a short period of time until eventually their health kind of nosedives because they're not receiving the treatment. This again is the placebo effect, where both groups respond favorably whether or not they're receiving the treatment. Just kind of an unfortunate aspect of some experiments. In some cases, the placebo effect can lead to a little bit of a bias. And let's talk about bias. This is a story about Clever Hans. Clever Hans is in fact the horse in the photo. He was a German horse that could perform math, potentially read, as well as kind of spell. However, his big claim to fame was being able to perform math problems. Now, I know what you're thinking. Rachel told me to be skeptical of claims that I hear, and I'm glad you're skeptical that potentially this horse is not able to actually do all the things I'm telling you it could do. Now, at the time, people wanted to believe that this was true. They wanted to believe in a world where a horse could do a math problem. Unfortunately, the horse could not do math. However, Hans was still very clever. It's just that if the person asking the question was hidden from his view, or if the person asking the question didn't know the right answer to the math problem, then Hans wouldn't be able to tell you the answer. It's because the horse was not doing math, he was reading body language. So still a very clever horse, just not in the way that we would have maybe wanted him to be. However, this is a form of bias. People wanted to believe that that was the answer, even though we find out later it was not. Biases are something that is very important to watch out for when it comes to experiments. A bias is a systematic error that leads to results or conclusions that are not an accurate reflection of the true underlying data. So, a bias essentially sways the data in a certain direction, even if that is not the true direction. In order to escape a bias or to build more confidence in your results, you can design your experiments as a blind experimental design or a double-blind experimental design, and then on top of that, add in randomization. So a blind experimental design is the experimental subjects will not know which treatment they're receiving. So either they're in the experimental group or they're in the control group. Either way, they don't know if they're receiving the treatment or not. That way we can get rid of that bias, however you may see the placebo effect. In a double-blind experimental design, neither the subject nor the experimenter will know who's receiving what treatment or not. So this ensures that the experimenter themselves is not creating a bias to their treatment group versus their control group. And then, of course, adding in an extra precaution is randomizing the subjects and the control group, the experimental subjects and your control group subjects as to who's receiving what. Unfortunately, in some cases, certain medical treatment studies will choose people who are less sick than others in order to make it seem like their product is working a lot better than it actually is. That is a bias, and it's also lying. So that's why randomization and a double-blind experimental design is probably your best bet. So that is the best way to perform an experiment in order to make sure that you do not have a bias and we can have a lot of confidence in our results. Another way to ensure confidence and to get away from a bias is to be able to repeat an experiment. So whether that's because of a few things, that the materials and methods were written really well in, say, the paper that we present for our experiment, or the data is particularly fantastic. When it comes to repeating the experiment, you get the same data set or a similar data set over and over. This replication is going to help ensure that confidence within the results. Now we kind of have moved into talking about the parts of a paper. So replication by good materials and methods, as well as good data, that's important. But also displaying our data is another important aspect of any paper. There are a handful of visual displays of data that can help us understand the patterns and relationships that we might see within the data itself. A handful of these could be like a bar graph, a line graph, or a pie chart, things I'm certain you've probably seen and worked with before. Now, I'm sure you've probably worked with some of these visual displays before. However, just to make sure that we know the parts of a visual display, let's go through it. The first is the title. This is going to give your reader an idea of what it is that they're looking at when it comes to your data display. It's a very important part and should go on every single visual display of data. On the tall axis to the left, that is the y-axis. That's where your dependent variable is going to go. So you're going to have some type of title explaining your dependent variable as well as some units that make sense. You don't want ambiguous scales on the side there, or along the bottom, the long axis, which is the x-axis, and that's where the independent variable and a title as well as a unit of scaling is also going to go. We are going to travel down a little bit of a journey where we end up at something called bad science. So there can be misleading displays of data, and you might be asking, well, how can data be misleading? Well, making your scales or labels kind of ambiguous or difficult to understand or read, as well as having incomplete information on how data was collected, so bad materials and methods. This makes it difficult for someone else to come in and try to replicate your methods to get similar data. Another kind of big thing is unknown or unreliable data sources. If you look at research papers produced by potentially AI, AI gets kind of confused and it will not produce actual research papers that support a research paper itself that it's writing. It'll make up sources to support itself. Now, you might be thinking, yeah, but that's AI, right? I would just check it. Nope, sometimes people do this too, and that's not okay either. Or using a data source that is not very good. Now, Wikipedia is fantastic for 100 reasons. However, I would not cite Wikipedia, but I would use Wikipedia as maybe a jumping off point to find data that is reliable, right? Other things that can lead to displays of data being misleading is biases or hidden assumptions, or maybe insufficient or inappropriate context for the data representation. Now, ways that help us kind of support our data is statistics. Now, don't groan, statistics are very important. Statistics are a set of analytical and mathematical tools, usually in the form of tests, designed to help researchers gain understanding from the data that they gather. This is incredibly important in order to take your data, plug it into some statistical test that is appropriate, and that can be another way that some data sets can be a little bit ambiguous, is using the wrong statistical test. That is a no-no. But for the most part, most people use statistics the way they're intended to look for relationships, patterns. So searching for the greater difference and the smaller the variation between two groups gives us more confidence that our outcome of our data did not occur from chance. So I mentioned chance earlier in this lecture, and that is the hope and the goal that we can support our hypothesis that there is a relationship between this thing affecting this thing. Statistics are also important in identifying correlations. So this is kind of a fancy word for like a pattern. However, a positive correlation means that one variable increases, the other variable will also increase. The opposite of that is a negative correlation, where one variable decreases, the other variable will also decrease. That being said, sometimes people look at correlation as if every time correlation occurs, every time you see a pattern, that there is causation there. And that is not the case. You've probably heard the phrase correlation is not causation. I want to give you an example. When we look at ice cream sales on the coast, let's say in California. We see that there is an increase in ice cream sales, and it matches an increase in shark attacks. Does that mean ice cream causes shark attacks, or sharks cause ice cream sales to increase? There is a correlation there. However, there is no causation. Ice cream does not affect shark attacks and vice versa. What we're seeing is that usually during that data collection period, it's probably warmer months, maybe the summer, and more people are going outside, maybe to the beach, getting an ice cream, and other people are also using the beach to go in the water. More bodies in the water, more chances of encountering a shark. So correlation is not causation. Other misleading forms of evidence that can obscure the truth may lie within two categories. The first is pseudoscience, where pseudo means fake. So we're saying that this is scientific-y kind of sounding things, but it's not actually science. It's not based in any methodical, scientific studies. So maybe the phrasing, 4 out of 5 dentists approve of this toothpaste. Well, that's not a statistic. It sounds kind of sciencey. However, it doesn't make any sense. Did you only ask 5 dentists? Why did the one dentist not like the toothpaste? What were the credentials of the other 4 dentists? Have they been using the toothpaste, or are they suggesting it for their patients? What does that actually mean? You can't tell me and they can't tell you because it doesn't mean anything. That is not a statistics and that is not science. Another example is product phrasing. So the product vitamin water, which I'm sure is lovely. I've never drank it. I do see students drink it frequently and enjoy it. I assume it is mostly water with some type of flavoring and maybe added vitamins, but I don't actually know. Why is it called vitamin water? Is it that important that it needs to be called vitamin water as opposed to really good water or something like that? But they also have titles on the flavors like Revive and Defense. Does that mean it's really important and integral to our life to be able to have that vitamin water? Because again, I've never drank vitamin water. Am I going to be okay? I don't know. That's where that pseudoscience, that phrasing that makes you think it's more scientific than it is, comes in. Our other misleading thing is anecdotal observations. This is making conclusions about relationships based on only a few observations, so very few observations leading to a conclusion. A good explanation of this would be like your horoscope. I myself am a Gemini. I like to read the Gemini blurbs when it comes to my horoscope, and I usually find I can relate to being a Gemini when I read about Geminis. However, that being the case, I don't really read the other horoscopes of any other person. I just stick to reading the Gemini one. But if I took the time and I read all 11 other horoscopes, I might find I relate at least a little bit to all of them. It's because I'm making an observation based on a conclusion based on a small observation. I'm only reading the Gemini one. Of course, I'm going to find something I relate to. That is anecdotal. Now, a really good example of very bad science and poor scientific thinking that led to years, decades of very dangerous behavior, and we're actually seeing a particularly bad fallout from this in current times, and that is the claim that vaccines cause autism. The short answer here is no. It does not. And there is no link, and there has never been a link between the two. The reason that this gossip or rumor got started is that in 1998 there was an article that was released that stated that there was an implication that there was a link between the MMR vaccine, which is measles, mumps, and rubella, and autism, that it caused autism. However, the doctor who we will not name here, he was being paid off by a rival pharmaceutical company to basically run this MMR vaccine through the mud so it would be taken off the shelf and a different vaccine would take its place. So, since 1998, now we're going on almost 30 years here, no one has been able to repeat this data using the materials and methods stated in the article. No one's gotten the same data at all for 30 years. So one of the biggest issues is that there was only 12 children in the study at all, and 8 of them had already shown evidence of being diagnosed with autism. So, how do we know that there's no link between vaccines and autism? Well, the best thing I can tell you is that Denmark, the whole country, has been doing a study for 30 years since that article came out, looking at over 650,000 children, and there is no link between vaccines causing autism. There's also been hundreds of other studies since then. However, we still see people reporting that, oh, my kid, he developed autism after I got him a vaccine, or he started developing all these conditions after he got the vaccine or a vaccine, whatever it may be. And once again, I want to draw your attention to ice cream sales and shark attacks. If you see one thing happening after another thing, you might think there's a correlation, but remember, correlation does not mean causation. So if I were a mother and I gave my child a vaccine, maybe for the flu, or maybe it is the MMR, or maybe for chickenpox, either way, I get them a vaccine. And then a week later, they start showing signs of autism and they become diagnosed with autism. I might think that there is a correlation between the two. And that is what we are seeing nowadays. The other thing that we are seeing is people are scared to vaccinate themselves or vaccinate their children, and that is leading to a rise in measles and whooping cough, as well as other diseases that initially we thought they were eradicated because people were getting vaccines. But now they are not, because they are afraid vaccines will cause autism. But it doesn't. There are limits to what science can do. Science is pretty cool in a lot of ways. However, there are limitations. First and foremost, remember that the scientific method is a physical process. It is empirical. Science is not going to give us moral statements or insight into ethical problems, whether that has to do with the presence or absence of God, or maybe the presence or absence of goodness versus evil, things like that. What science does give us is sometimes products, so something like technology, which is not science, it's actually the application of research, but especially when it comes to things like prosthetics, or medications, or things like, you can think of pacemakers. That's technology that's come a long way and ensured that individuals are able to survive for a lot longer than they previously had with those same conditions. So science is limited in what it can do. However, there's a lot of cool directions that we've been going with science. So moving on to what is life. This is going to be a very important unifying theme within all the chapters pretty much that we're talking about. So what is life? How do we define it? How do we distinguish it from non-life? Because the opposite of life is not necessarily dead. It just means something that is non-living. According to biology, there are a handful of characteristics that lead to something, a species, being classified as living. The first is having a cellular structure. So this is a complex ordered organization consisting of one or more cells. We'll actually talk about that a little bit more in chapter 4. The organisms, so again, we are talking about a species here, being able to metabolize, so transforming something into energy, whether that is transforming light, like a plant does, or transforming a burger into taking those calories and converting it into energy so you can move your muscles or think properly when you study, right? The third characteristic is sensitivity and responsiveness to the external environment. That could be a response to temperature, light, maybe electricity. It could be a response to water or touch. It just kind of depends. So responding to an external environment. Homeostasis is the next characteristic, and I know that's potentially a word you've never heard of. Homeostasis is just the optimal internal state, and organisms that are living do have to maintain that optimal internal state, whether that's through controlling your blood pressure or your temperature, controlling your metabolism, things like that. It's very important. It's how you stay alive, right? There's a lot of different processes that are important within your body that keep you existing, right? The next characteristic is growth, development, and reproduction. Emphasis on reproduction. A species must be able to reproduce on its own in order to be considered a living organism. And last but not least, you need to have an evolutionary adaptation. So this is evolution leading to descent with modification over time. So your species must change over time in order to be considered a living thing. Now, one of the organisms on the planet that kind of spans whether or not it's living or non-living is viruses. Viruses can do quite a few things on this list. However, one of the biggest things that leads to the controversy of are viruses a living thing is they can't reproduce on their own. They have to hijack our cells or a host cell, not necessarily human, because other organisms can have viruses, plants can have viruses, but they hijack a host cell, basically deconstruct it, and build baby viruses out of the parts of the host cell. So they can't do that on their own, which is the biggest conflict of whether or not a virus is living or non-living. And with that, guys, that is the end of chapter 1. Congratulations, you finished the first lecture. From this point, you should be working on homework. You should be studying and holding on to any pieces of information that involve chapter 1.